Welcome to Quindecim. This time I'll be talking about Chapter 12, DNA and RNA. Now, let's get started with... Lesson 1 starts off with Griffith and his experiments. Griffith isolated a strain of pneumonia from mice and examined how the two strains grew in different ways. With these two strains, he injected the harmless strain into mice, and nothing happened. When he injected the disease-causing one, it did what was expected, and it gave the mice pneumonia. After this, he heated the bacteria, then injected it into mice. The mice survived, and he discovered that the pneumonia isn't caused by chemical poison released by the bacteria. After doing this, he did another experiment in which he mixed the heat-killed bacteria with the harmless, then injected it into mice. He was surprised that the mice ended up dying from pneumonia. After looking closely at the mice, he discovered that the heat-killed bacteria passed their disease-causing abilities to the harmless strain. He called this transformation. The lesson then explains that Avery and other scientists discovered that DNA is nucleic acid that transmits and stores genetic information from one generation of an organism to the next. After this, it talks about Hershey's and Chase experiments and studies on viruses. One of these viruses being bacteriophages, which are viruses that kill bacteria. With these bacteriophages, they use radioactive markers to find out what the genetic material of the bacteriophage was. After the experiment, they concluded that the genetic material of the bacteriophage was DNA, not protein. Next up, we get a whole bunch of information about the structure of DNA, which basically says that DNA is composed of 5 carbon sugars called deoxyribose, a phosphate group, and a nitrogenous base. Nitrogenous bases are classified into 2 groups called purines and pyrimidines. Purines are adenine and guanine, and pyrimidines are cytosine and thymine. Chargaff has a rule that says that cytosine goes with guanine and thymine goes with adenine, aka base pairing. Rosalind Franklin did some X-ray stuff with DNA and found that the patterns she found didn't really reveal the structure, but hinted that it had a spring shape. 2 guys named James Watson and Francis Crick looked at the patterns and concluded that it was in a double helix structure. Now that this lesson is finally over, let's get to the next one. Now let's get started with what chromatin is. Chromatin is when DNA and proteins are tightly packed together. Chromatin are made of DNA that is tightly coiled around proteins called histones. Next we have an explanation of what replication is, which is a process where DNA duplicates its DNA in a copying process. In DNA replication, the molecules separate into 2 strands, then produce 2 new complementary strands. Each strand of the double helix of DNA serves as a template for the new strand. DNA polymerase also plays a hand in replication because it polymerizes individual nucleotides to produce DNA. Next we have RNA is basically the same as DNA, except it has ribose instead of deoxyribose. It's usually single-stranded and has uracil instead of thymine. There are 3 types of RNA: transfer RNA, messenger RNA, and ribosomal RNA. Messenger RNA serves as messengers for DNA to the rest of the cell. Ribosomal RNA is a component of ribosomes. Transfer RNA transfers amino acids to the ribosome as it is specified by the messenger RNA. Transcription is a process where RNA molecules are produced by copying part of the nucleotide sequence of DNA into a complementary sequence in RNA. RNA polymerase is basically DNA polymerase, but for transcription in RNA. The enzyme binds to regions in the DNA called promoters, which have specified base sequences. RNA is then edited and has introns removed, and what remains is exons. Genetic code is a set of 4 different bases: adenine, uracil, cytosine, and guanine, put into a sequence in sets of 3s. Messenger RNA then has its messages decoded in a process called translation, which takes place on ribosomes. Now, on to the next lesson. There are two types of mutation: point mutations and frame shift mutations. Point mutation is when a single point of a DNA sequence is changed. Frame shift mutation inserts an extra base. Point mutation isn't always fatal, but frame shift will always be fatal. This lesson doesn't really have any other relevant information, so let's just go to the next one. Operon. Operons are a group of genes that operate together. Since these genes must be expressed in order for bacterium to be able to use the sugar lactose as a food, they are called lac operon. Lac genes are turned off by repressors and turned on by the presence of lactose. Eukaryotic genes are controlled individually and have regulatory sequences that are much more complex than those of the lac operon. Hoax genes are genes that control the organs and tissues that develop in various parts of the embryo. You can kind of see that I didn't really put it there because all the examples were like bugs, and I don't like bugs. Well, that's basically all the information that is of any real importance in this lesson. Thanks for watching, and I'll see you in the next video tutorial.